All right, the citric acid cycle, also known as Krebs cycle. There he is. So this is going to be occurring, as we know, the powerhouse of the cell, but specifically within the matrix of the mitochondria. So again, the most inner part of the mitochondria itself, inside of both of the phospholipid bilayers that make up the mitochondria's outside part. New molecules that we haven't seen yet so far in cellular respiration are GDP and GTP, which are very similar to ATP. So again, diphosphate and triphosphate, but instead of adenosine, it's guanosine, diphosphate and guanosine triphosphate. So they're going to act like ATP, and in fact, as soon as we make them, we're going to convert them into or use them to produce an ATP anyways. So just think of them as almost ATP, but we've just got to use them to get to our ATP. And we're going to be using our other electron carrier. So far we've seen our NAD+, which picks up the electrons to form our NADP, which is our energized electron carrier. And this is FAD, which picks up electrons and becomes FADH2. And again, we're not mentioning the hydrogens involved in that conversion process. We're just referring to FADH2, just as NADP is. They are electron carriers. All right, we're going to start the citric acid cycle with our acetyl Coa. So this was, again, the result of our pyruvate oxidation, the oxidation of pyruvate. The pyruvate itself came from glycolysis, which started off with glucose. So we took glucose, we turned it to pyruvate. During glycolysis, we took pyruvate and oxidized it into acetyl Coa during pyruvate oxidation. Now we're going to take that acetyl Coa and we're going to run it through this cycle. And as a cycle, it really doesn't have a beginning or end. It's just continually running. So as we enter the cycle with our acetyl Coa, it's already in process with this 4-carbon molecule here called oxaloacetate. So we take our 4-carbon oxaloacetate and we add in our 2-carbon acetyl Coa plus some water, and we end up forming this molecule called citrate, which is a 6-carbon molecule. So the first step involves adding acetyl Coa to this previous existing oxaloacetate, which is already in the cycle, and being as this is a cycle, we're going to want to get back to oxaloacetate. So this process will take us through a series of events. We're having things added and removed, but we have to end up back at oxaloacetate, otherwise it's not a cycle. So citrate gets produced in the first step. It gets converted into isocitrate, so just another isomer of citrate. And then the step that we first see a change in the carbons here is when we take our isocitrate and turn it into ketoglutarate. It's alpha ketoglutarate, in case you're wondering. But the important part is we have this 6-carbon isocitrate, and you'll notice that we are now down to 5 because we have lost a carbon in the form of carbon dioxide. This is where we get another one of those 6 carbon dioxides that are generated during cellular respiration. Also in this step, we generate an electron carrier. We reduce NAD+ into NADH. Remember, reduction is gain. NAD+ gains electrons and becomes NADH, which means isocitrate is being oxidized. It's losing electrons on its way to form this alpha ketoglutarate. Process happens, or a similar step happens again. We produce another carbon dioxide, and so our carbons drop down to 4 now when they form this acetyl Coa. Also, another electron carrier is generated. So actually, let's put that in red here. So there's an electron carrier, there's an electron carrier. And so we're going to put them off to the side. We're going to use them later. More on that later. These carbon dioxides, though, are being lost, and they're waste products. We'll get them out of our cells and breathe them out into the atmosphere. And that is where we are so far. We've started with this 6-carbon citrate here after we've added acetyl Coa to oxaloacetate. They came together, formed citrate, and we have now lost 1, 2 carbons here, and we're down to a 4-carbon molecule. This step here, succindate here, is another 4-carbon molecule, and it should see as we go through, we've got to have this 4-carbon oxaloacetate, so we're done losing carbons. But what we can do is that we can take, essentially this CoA is being removed here, and it can be used to generate a GTP, which is then used to generate an ATP. Get a different color in here. So this ATP is generated, and we can then use that to do some other processes. This is our energy generation part of the Krebs cycle. We get 1 ATP per acetyl CoA that enters. And don't forget, we've made 2 electron carriers so far. Another electron carrier that we can produce is our FADH, when FAD plus is reduced into FADH. Remember, reduction is gain. It is filling up that electron carrier, and that electron carrier can join the NADHs over here. So we've made 2 NADHs and 1 FADH so far. Another conversion, we were adding in water here, and we end up forming our malate. And our malate is almost on the way back to the next step. We'll take it back to oxaloacetate, but we do generate one last NADH, so we are oxidizing the molecule in the middle here once more, and by doing so we can reduce NAD plus into NADH, and we have our 4th electron carrier. We have 3 of them that are NADHs, and we have 1 FADH, so we've produced a number of electron carriers. We've produced an ATP molecule. We've produced some waste carbon dioxides, 2 of them there, right? It makes sense if we started with 6 carbons and we've gone around the cycle and now we have 4 carbons. You got to get rid of 2 carbons to do that. Since it's a cycle, we're going to put back in 2 carbons in the form of our acetyl CoA and run the cycle again and again and again and again. And so each time we turn the cycle, we can do 1 turn per acetyl CoA. We get out some carbon dioxide, some electron carriers, and an ATP. Summary of it, remember, for every glucose, you're running this cycle twice. You have 2 pyruvates and therefore 2 acetyl CoAs, and therefore you can run the Krebs cycle for 2 spins. In, we have oxaloacetate, which is already there. Acetyl CoA gets added in. Our unenergized energy carriers, GDP is going to be energized, and it is going to be made into GTP, which is then used to make ATP. So ATP is the goal there. We're making 2 of those. Our empty electron carriers get turned into full electron carriers. And if you run the math on this, out of this 1 glucose that we're doing, we get, importantly for us, 2 ATPs. We get 6, remember 3 per cycle, 6 NADHs, 2 FADHs. Those are our electron carriers. And then to work the carbons out, also note that we are losing 4 carbon dioxide, 2 per spin, which makes sense because we're putting in 4 carbons, so we have to get 4 carbons out. Each acetyl CoA has 2 carbons. We run it twice per glucose. That's going to produce 4 of our carbon dioxides, which, factoring in the 2 carbon dioxides that we made during pyruvate oxidation, that would be a total of our 6 carbon dioxides. And if we think of the overall equation for cellular respiration, that is it for carbon. We have converted all of the carbons from glucose into their products of the reaction. So let's look at the overall written part here. Again, oxaloacetate, acetyl CoA is the sort of one that we're entering into the cycle with. Oxaloacetate is already there. These sort of empty versions of the energy electron carriers and energy carriers are there as well. Realize I'm skipping over the GDP to GTP transfer. And then again, what we want to focus on, what we're getting out of it, we get our electron carriers and our ATP, and then our waste carbon dioxide, plus the oxaloacetate, which is run through the cycle again. Looking at the carbons quickly, we have 6. We're going to be losing 1 of them here, and so we have our 5 carbon as we're losing this first carbon here. We lose a 2nd carbon there, producing our 4 carbon compound that is going through, and they all have names. Feel free to memorize them if you like. We have one electron carrier generated here, one NADH, two NADH, and our last run up at the generation of oxaloacetate, that's 3 NADH electrons carrier generated, plus 1 FADH2 generated, plus our ATP generated, and again this 4-carbon compound sticks with it until you get this 2-carbon added to this 4, giving you back to our 6. Looking at the overview of the glucose, let's look at the 6 carbons that went in. All 6 of them made it through glycolysis, and so we started glycolysis with the 6-carbon glucose that had been split into the 2 pyruvates, but no carbons have been lost. They've just been split into 2 groups. We then ran the pyruvate oxidation, also known as transition reactions, and in that case we did lose 2 of them. So we're down to our 4 carbons from our 6 that enter as glucose, 6 leave glycolysis as 2 pyruvate molecules, but at the end of the pyruvate oxidation, 2 of them are gone, so there's our 2 CO2s that leave the system and our waste. 4 carbons continue on, but then all 4 of them are converted into carbon dioxides during the Krebs cycle, and again we're doing 2 spins of the Krebs cycle to get those 4 carbon dioxides, and that accounts for all 6 of the carbons that entered our cellular respiration as glucose.